But it is my pleasure, and thanks to all of the APG team for working really hard to still allow me to kind of connect in for these next few minutes and really just kind of set the stage for the headliner. So nobody goes to the concert for the opening act, and today that's what I am. But I think it's super appropriate because today is World Soil Day, that we're going to spend the next 45 minutes to an hour or so talking about soil, really, and some of the challenges and the pathogens in the soil that affect pulse crops, specifically Aphanomyces and the other root rot complexes. And so kind of like I said, I'm just going to give you really quick a little bit of an overview because I don't have much of a voice and it won't last long. So when we think about the last 8 or so years, that's kind of when the word Aphanomyces got introduced into the pulse world. And Syama will have some specifics, and you know, if you've seen her present before, we've kind of seen this slow march as we identify kind of where Aphanomyces has been located. And you know, if we think back, often it feels like we're looking for that straight line of success. And I think, you know, kind of the reality, and we can probably all identify, is that we're somewhere in this mess in the middle. But a couple of the things that I want to bring forward today is that we are on an upward trajectory, even if it doesn't feel like it, and just kind of give you a bit of the strategic thought process behind how Alberta Pulse has invested in this space. And so, Colin, I'll get you to advance the slide, please. Like I mentioned, 2013 was kind of our first introduction, and we really started out working at some soil surveys. And bottom line is really, and Syama is going to kind of come behind me and sweep up all the pieces and redirect where I've gone a little bit wrong. But if you have soil and you're planting crops, there's a good chance that you're at risk or may come in contact with the soil pathogen that we know as Aphanomyces, as well as a host of other root rots. So Fusarium, Rhizoctonia, Pythium, you know, kind of those complexes of diseases that we're also familiar with and have become, you know, really intimately aware of over the last, of course, number of years. So kind of from a strategic standpoint, when we're investing in research in this space and over this time frame, the Pulse Corps have invested right around $3 million in root rot focused work. And that's a big chunk of change. And sometimes it's hard to feel like we've made progress. And so just kind of if we take a little bit of a step back, I would say that there's 3 main areas of investment. And the first one is looking at the plant itself. And so when we think about investment into the plant, really what we're talking about is resistance and working towards building those resistant varieties. And so there's a number of different projects. Every breeding program now screens for this. Syama's very much involved in that. And, you know, kind of it involves actually planting out varieties in infected soils, in greenhouse soils, and just seeing and observing what happens. And we call kind of that physiological resistance. And I think, you know, if we think about some of the lines and stuff that we've got, we may sometimes see some varieties doing a little bit better than others. But kind of the sad reality is at present, we really haven't seen any of that physiological resistance either transfer to the field conditions or show up in the genetics. So there's been a little bit of progress in lentils where we have a lot of what we call wild accessions, which is just looking kind of at all of the lentils that are grown, not necessarily cultivated, and seeing if we can find some resistance in that. And I think we're making some progress in that space. Peas are a little bit more challenging, and so we're leaning in and actually looking at some really novel, outside of the box ways that may be bringing in resistance. And you can read between the lines however you like with that. But you know, kind of the point is that we're here for the long game. And even though maybe some of these plants with novel traits aren't accepted currently, there is a chance in the future that they will be. And so we're very carefully exploring some of those potentials and possibilities as we really lean into looking kind of for that resistance, that we can be confident in planting varieties into the field. I'll get you to advance the slide, Colin. Kind of the other general area that we actually realized we were really unprepared for originally was we really didn't know much about the disease itself. And so, you know, Fusarium and Rhizoctonia and Pythium, those diseases have been around for a long time. We're a little bit more familiar with them, but Aphanomyces was a bit of a black box at first. It is also a different beast, and Syama can go into the specifics, but it is a water mold. And that tells us a lot of things about it. It needs moisture to move. And so it actually kind of looks like a little sperm cell with two tails, which means that it swims. And so moving through the soil, it really does. And if you kind of observe pathways through your fields of infection, you can see that it's moving with the pathways of the water. So that's really key. And if we think about kind of the last number of seasons where we should have maybe thought there was a field that was infected, where it was really, really dry, we didn't see a lot of disease. And one of those reasons is because of the water. One of the things we also learned early on is we were misidentifying it. So we were getting a lot of false positives. And so one of the things that Syama focused on right at the beginning was accurately detecting this. And so we've done a lot of work, and now there are labs that can confidently diagnose a plant with Aphanomyces. And we have also worked on looking at the number of spores per gram of soil that actually cause disease. And so you can, in a planned rotation, send a soil sample away to a lab, excuse me, and get it tested for the number of, for presence or absence of Aphanomyces spores. And kind of their tipping point is that 100 spores per gram of soil, at which point we would say, yes, you have a risk of getting a pea or a lentil crop infected with Aphanomyces. And so lots of little learnings along the way. One of the things that we're currently working on, and I'm sure Syama will touch on, is how long do these spores live in the soil? And we're kind of examining that for a number of reasons. We've talked a lot about really our only defense right now is to take a pause from some of these crops. And so, you know, if you're on a 1 in 4 rotation with peas and you've had Aphanomyces, we're suggesting that maybe you want to go as far out as 8 years. And one of the reasons for that is we just don't know how long the spores live. So another kind of tidbit of information is Aphanomyces is what we call a saprophyte. And what that means is it can live and exist on any kind of plant material, but it will only reproduce on an acceptable host. And so in the pulse world, that's peas and lentils. And so that's one of the reasons why, you know, we want you to increase rotations and maybe not remove pulse, but choose a pulse in that 4th year that is resistant. So a faba bean or a chickpea or a soybean. Lupins also appear to be resistant to this disease. So, you know, we're making learnings all the way along, and I know that Syama will touch on some of those. The third and final kind of area that we're investing in, and Colin, I'll get you to advance the slide again, please, is kind of the interaction. So if we think about a disease triangle, we need the host, which in this case is a pea or a lentil, and we need the pathogen, so whether that's Aphanomyces or Fusarium or Rhizoctonia, and then we need the environment. And we talked a little bit about, you know, the need for moisture already. Heavier soils are a little bit more susceptible just because they tend to hold on to that moisture longer. But can we alter the environment? So this means soil amendments. This means looking at different crop rotations. This means looking at protection products. And so those are kind of the 3 pillars where we're really, really focused on research investment in this space. Leanne mentioned in her portion of the presentation this morning that, you know, kind of as a collective, we're really trying to work hard at formalizing what we're going to call the root rot network, which is really just a nice way of saying that, you know, here in Alberta, in Saskatchewan and Manitoba, we've all been working fairly independently in this research space. You know, we share a lot of the same scientists and extension materials, etc. But we've really started to formalize this group And the concept behind it is that instead of having 3 engines running on parallel tracks, we're going to put all our engines on the same train and really make efforts to leverage each other's funding, and to really kind of hopefully propel ourselves forward, to really solve this challenge. It's been called the Achilles heel of post production. And I think it's a rare occasion now where, as a post producer, you haven't been impacted in some way, in some year, by some of these root rot diseases. So I'm going to get Colin to advance this slide. And I know sometimes it feels kind of, even as we think about, our farm and we're planning our crop rotation for next year, the field, one of the fields that we had slated for peas is a bit heavy. There's some gumbo patches, and we're really kind of scratching our heads with what's the best way to move forward and manage this piece of land, knowing that we want to keep good crop rotations, etc. And I think we can kind of all identify with the top of that photo where, in this space particularly, it kind of feels like we're climbing a mountain. But I think if we break it down and kind of do a bit of a survey of where we've been, we'll actually see that we're on a staircase, and we are nowhere near the top of the stairs yet. But we've certainly made some progress. And I think that's, while our toolbox is still fairly light, there are some tools in there and some things that we can do to empower our decisions. And so I'm going to close kind of my real high-level brief overview with that, and invite Syama to come forward. And Syama is no stranger to any of us, and has certainly become a real leader across Canada and even North America in this root rot space. She's got her hands in nearly every active project that's going on, and so I just encourage you to ask her the hard questions, and just give her a warm welcome.
Oh, that's a really tall. I can get it lower. Hopefully you guys can hear me. I have a problem a lot. Oh, there we go. Okay, there we go. Hey, thank you, Jenn, for that very warm welcome. I know we got a lot of pressure on me to kind of get this disease solved. Yeah, and I just wait for my slide to load. There we go. So I'm just gonna jump right in and start with what Jenn talked about, and just have a list of projects that APG has funded since 2013. So, next year is our 10-year anniversary of Aphanomyces. I don't know if we want to plan a party for that or not. And really what we started looking at 10 years ago was that we thought we were dealing with a fusarium root rot problem. So that was really what my first research project was, figuring out what fusarium species we were dealing with. And then there was that nice surprise of Aphanomyces. So then the next project was moving into looking at, okay, well, how widespread is Aphanomyces? And then the next kind of 4 projects that I've highlighted there in bold is starting to look more at some of management strategies, kind of a combination of both field work and a lot of lab work. So I'm focused on trying to summarize those 4 really big research projects in the next 25 minutes, half an hour. And just to keep it short, to focus mostly on the field work that we've done and not all of the lab work. I could be up here for 3 to 4 hours, but Nevin would probably pull me off stage, I think. And then there's right at the bottom there's actually 2 new projects we're just funded, and those are really where Jenn talked about really trying to figure out what's happening with the pathogen. Those 2 projects are really delving into some of those really more finer details of the pathogen. And with APG's money, we've funded a lot of projects, but we get a lot of matching projects as well. So usually whenever I apply to APG funding, I'm also applying for matching funding from Saskatchewan pulse growers, And then also usually that comes with matching funding from WGRF, ARDAR, ACIDF way back in the day. So we get quite a good network of funders that can help fund this research. So now I'm going to get into research, and I want to say that this is about 4 years of research summarized into one slide. And this is really our first project that I talked about, where we are trying to figure out what are our pathogen problems. And really, we're looking at kind of a whole suite of these root robbers. I call them root robbers because that's what they're doing. They're stealing the roots from the pea plants. And our number one problem, as we all know now, is Aphanomyces buteiches. It's part of our lexicon, whereas I think one of the first times I got up and talked in farm tech, we actually had everyone practice how to say Aphanomyces because it was a totally new player on the board. So now we all know Aphanomyces, and I like to think it's our big bad. It's punching its way into that root. It just wants to destroy those roots. And then it pulls along its little kind of helpers there, the supporting players, some of these fusarium species that can cause a root rot on their own. But usually it's Aphanomyces that cause the most damage. And then at the bottom, we have the little kind of bit players that you're just sort of hanging along for the ride, but they don't really know why they're there. They're just, hey, some free rotted roots, let's get in there. And then what really helps along is those wet soils, pulse history, and tight rotations. And then just kind of an overview of what we saw within those 5 years of that first project, and I think everyone's seen this map by now, is really Aphanomyces is present anywhere we see pea or lentils grown. And down here in southern Alberta, we can see that we get a lot of positive fields over those number of years. And if we break down what we saw in Alberta, since it's not moving. Now too far. Okay. So this is just really a summary of Aphanomyces that we've seen in Alberta since 2014, along with precipitation. Ben talked about how important water was in this disease. So we see high years, high root rot, high water years, high root rot. Those drier years, you can see that the root rot levels are really decreasing. 2015 is just a bit funny because that year we went out specifically looking for Aphanomyces. It wasn't really a random survey. It was like, if you have a root rot problem, call us, we'll go and look. So we found a lot of fields with Aphanomyces, but when you actually look at that incidence, it's quite low, which is usually what we see in dry years. And then 2016 was another wet year, incidence very high. So here we're looking at with all the fields that had root rot, 6 out of 10 sites in a field would be positive. Then we had a couple of dry years, 2017 to 2019. We're going like, oh, we solved the root rot problem. Then 2020 ended up actually being quite a wet year, and then we see that it jumps up right away. And we're still catching up from COVID, so I don't have the 2021 data yet. And then lentils, very similar pattern. The big difference is that dry years made a really big difference in lentils. So in 2016, that wet year, we saw it about 50% of the fields. 2017, 2018, 2019, we didn't even find it in lentils because it was dryness. And back to 2020, we're back up to 50% of lentil fields. So what happened in 2022? This was a really odd year because when we look at the rainfall patterns, and this is for Tabor, those first May to June seemed like we were in for another really dry year. And then all of a sudden, in the middle of June, right down here, we got this huge bump of moisture. And then it got dry, and then actually we got, mid-July, another big rainfall. So that we ended up with the kind of annual, or the totals from May to August is about 280 millimeters total rainfall. Compared to that was 66 millimeters in 2021. So a huge difference. And then I'm just going to show you as well from Red Deer, because we have a trial up there. So very similar, dry and extremely wet. And they actually ended up with, can't quite see the bar, but I think it was about 320 millimeters of rainfall over that growing season. It's always a bit wetter there. So in 2021, they had about 244 millimeters of rain. Now, I don't often like to say I told you so, although don't ask my husband if that's true. But this is But I tweeted in mid-June this year saying we're all really happy for this rainfall, but watch out for Aphanomyces. It's coming in 2 to 3 weeks. And that's exactly what we saw. We looked at kind of the survey data. Mike Harding's group goes out and does this now because we have kind of land access issues because of the Trespassing Act. Mike's group goes and does the surveys and then gives the routes to us. What he did find was that, yes, indeed, 2022 was a very, very tough year for root rots, and it's why it's on everyone's mind again. Where we saw about 95% of the fields had root rot and an incidence of about 55%. We haven't analyzed all those data yet to actually see how many of those were positive for Aphanomyces, but I'm guessing we're about the 50 to 60% mark again this year. For lentils, we didn't go out and do surveys, but people submitted samples to both myself and Mike and tested those, and about 60% were positive for Aphanomyces. So again, we're kind of sitting at that really wet year where we see 50 to 60% fields are positive. The other thing that was kind of interesting that we did this year was look at Aphanomyces on processing peas. Now I know processing peas aren't a pulse because we eat the fresh peas rather than the dried peas, but it's exactly the same plant. So we wanted to go out and see what was happening in the processing peas. And so we surveyed a whole bunch right around July 14th, so a good 2 to 3 weeks after a lot of that rainfall. And what we did find is that they were all positive for Aphanomyces. And here we can see kind of the disease severity plotted against the levels that we can find in the roots, and you can see there's a really nice correlation. So the higher disease, the more Aphanomyces you have on the roots. Now the really interesting thing with processing peas is that they have a staggered planting date, pretty much from beginning of May all the way to the end of June. So my group surveyed the ones that were planted in May, and Mike's group surveyed the ones that were planted a bit later, and he actually found a lot lower levels of root rot. So sort of this question is, does planting date matter? And next year we're hoping to survey more fields and get that exact sort of planting date and use that as a proxy instead of doing a field trial with planting dates. Just look and see, okay, well, is a later planting, are you kind of changing that susceptibility period? Okay, and Jen, oh, this is what some of our processing pea fields look like. Those really brown honey caramelized roots, and you start to see some of that yellowing in the shoots. So Jen talked briefly about Aphanomyces being a water mold. And try to get it to load here. Oh, there we go. So yeah, the big problem with Aphanomyces that I think everyone knows by now are these spores, these pesky little creatures you're going to see come up 3 more times. Is that they just sit in that soil and they like to sit there until you get a pea crop and until you get water. So we had our pea crop, we had our water, and that produces what Jen was talking about, these little zoospores that can swim around. And the part of the problem is it doesn't take a lot of oospores to cause disease, because you can get 100 oospores that then are producing thousands to hundreds of thousands of zoospores that go and find those roots. And once they find the roots, they colonize very quickly and infect those roots. So you get that honey brown discoloration, decays the roots quite quickly. I have a new biologist in my lab, Shamila, she's sitting over there, and she's really looking at how fast this process can occur by kind of tracking it like every 2 days. And you can see within 7 to 10 days, you get kind of complete destruction of the roots. And then once they've eaten up all the nice juicy bits, you go back to having the oospores that sit in the soil. So there we go. Okay, so that's our kind of disease cycle. So now I'm going to move over and talk about some of the research that we're doing, particularly related to field crops. One of the big projects is this use of cover/rotational crops. This has been a bit of a thorn in our side. It's been a very tricky field trial to work with, and kudos to Alberta pulse growers for really going through a lot of amendments and allowing us to kind of change what our original plan was. At first, we were trying to look at using shoulder season cover crops. You know, peas come off fairly early, so we're like, well, can we get one of these like brassica green manure crops or an oat green manure crop after a pea crop and reduce ooze spores. Well, we did that in 2018 and 2019, which were so dry that we put those put the crops in, beginning of August, thinking, oh, we have 2 months to kind of get some growth, and they didn't germinate until it snowed. Basically, was the only moisture that we had. So we kind of pivoted and changed the plan, and we went into looking into more rotational crops and kind of full season cover crops or green manures. And we tested about 20 different treatments, just some, standard canola or mustard as rotational crops, some brassica blends as more of a cover crop green manure, oats as a seed or cover crop. Anyway, a big list of treatments that I don't want to fully get into. And then what we did was plant them in 1 year, and then the next year we'd put in the pea. So this is just a really quick summary of kind of what we were looking at. So rotational crops, either taken, just plowed into the soil as green manure, or taken all the way to seed. Some were just cover crops, some of those brassica blends that are supposed to have some sort of biofumigant activity. And then we looked at tilling or not tilling in those, the different things. And really what we found was that there was no effect of cover crops as green manure, and there was also no effect of tillage. There had been a lot of questions about whether tillage would help. And from this trial, we really found that there was no difference between the tilled and the no-tilled sides. The rotational crops had some interesting results. Really, we saw that there was no effect on disease severity. But when we looked at yield, and I'm just showing the results from the best site, which was Lacombe, because they had the best establishment of the rotational and green manure crops, we can see that some treatments actually did really well in boosting that yield despite having fairly high disease severities. So Caliente mustard, which has very, very high biofumigant abilities, actually worked quite well as a green manure. Fava bean, surprisingly, also we got a really nice yield boost either as a green manure or taken to seed. And then I think, I can't quite see over there, but I think that was oats as a green manure. So that was really encouraging. We didn't see to the same extent as our other two sites in Lethbridge and Saskatoon, because those crops didn't grow as well in 2019, so we didn't have as much biomass for 2020. And then the other thing we looked at here is the amount of Aphanomyces kind of left behind in the soil after these crops at 3 different time periods. So time period 1 was the pre-seeding, so before we planted the rotation crop in 2019. Time period 2 was after that crop was pulled off. So in the fall, we took soil samples. And then we looked again after the pea crop in 2020. And 2 interesting things came out of this. One is that the variant that we have in Lethbridge up here in red is actually quite different than the variant that we have in Saskatoon. So that's showing down here in blue. So just interesting that we have maybe really different strains between different locations. And then the other thing is that really we didn't see Aphanomyces increase until after that pea crop. And then you can see it go up in the soil quite drastically after you have a pea crop. So it's telling us that these rotational crops aren't really impacting Aphanomyces levels in the soil. So that's why we're not seeing differences in disease severity. But for some reason, some of them are giving us differences in yield. So there's something else interesting going on. We haven't quite figured that out yet. But when we look at the 2021 to 2022, there we go. We had such poor growth. 2021 was so dry. There's just a picture of what our fava beans look like down here in Tabor. So we just had really, really poor establishment of most of these crops. And then 2022 was so wet. We had such high disease. This is our disease severity for Lethbridge. We really saw no difference again. And then Lacombe, which is usually our best performing site. It's coming, there's a little animation. It's the first time that I've done research where we've come back with roots that were all rated a 7. 7 is the highest it can go. It basically means it's dead. So it just shows you like what a crazy year 2022 was. And we don't have the yield data yet to see if we saw any impact of that. And I'm going to move over, probably need to speed up because I had a lot to talk about. I'm going to talk quickly about this project we've been doing with the pulse clusters on rotational crops. So this is a bit different, where it's not just a crop sequence. We actually had like 1, 2, 3, 4, 5 years of peas in rotation. And with just cereal and canola as our primary rotation, but then adding in a pulse crop, chickpea at Tabor and Swift Current, fava bean in Lacombe and Saskatoon, and then soybean in Redvers in Saskatchewan, and then Morden in Manitoba, and just seeing how things went. And I think I presented this data last year at the Pulse Growers meeting, so just try to go through it quickly. Okay, and really what we found is nothing. No, really what we found is if we look from 2018 all the way down to 2021, our disease severity stayed really high. And then if you look at yield, so I have rainfall here. So we have our really dry, our really dry years in 2018 and in 2021. And then we had our wet years. We can see that yield is really depressed in those wet years. So like whoever thought that you'd be like, well, drought is better than rainfall. But when it comes down to root rot and Aphanomyces, we can see that it's really that wet conditions that are depressing the yield. And then in 2022, our trial was hit by hail. So actually, again, the first time in 10 years since we've been running field trials that we lost one to hail. But the disease severity that we pulled off before we were able to get yield was also very high. So just showing what Jen said, we're looking at 2016 to 2021, so really a 5-year break in 2022, we really haven't seen that disease severity come down yet. And then I'm just going to show similar results from Redvers. So this is in southeastern Saskatchewan. They tend to be a little bit wetter. And then actually we saw some encouraging results here in 2020 where we saw a really nice reduction in disease severity. We thought, oh, well, maybe, maybe some locations we get a break a bit earlier, but then back up to 5 years. And then in 2022, which was their really wet year, we saw that disease severity come right back up. So why that kind of 2020 was sort of the sweet spot for disease not developing, we haven't quite figured that out. But again, looking at those long breaks that we're going to need in rotations. Okay, so that brings us to those pesky ooze spores, and I think Jen showed this picture as well. And really our problem is these little creatures here with these really thick cell walls that just protect them from any sort of environmental damage. We have a heck of a time trying to keep these things alive in the lab. They don't stay alive for more than 6 months, but somehow in the field they're surviving for 8, 10, however many years, and we're trying to figure that out. And so that's one of the projects that we've been working on for the past year or so, is looking at how long does it take to decay. This is kind of a hypothetical ooze spore decay curve. If half of them died in a year, how long would it take you to get below threshold? So our threshold is here at about 100. So if you started with 1,000 ooze spores in your soil, then it would take you 5 to 6 years to get below threshold. If you started with 10,000, you'd see it takes a bit longer to get below threshold. But we really don't actually know what that half-life is in an ooze spore. So that's what we're trying to figure out. So what we did was grow up a whole bunch of ooze spores in infected roots, and then we also inoculated soil. We put them in these little cages. They went out and get buried in these little trenches in the field. Look something like this, a little flag, so we can go and pull them up. And then we pull them up every 3 months and say, okay, well, how are we starting to see a reduction in the ooze spore levels? And the way that we measure that is actually looking at a disease severity. And so what we found over the first year is their half-life is longer than a year. So basically, this is ooze spores on our infected roots that we buried in November 2021, and then we pulled out every 3 months. We can see that we have no reduction in disease severity. If those ooze spores were kind of decaying and dying, we would expect to start to see a reduction in disease severity. When we put them in soil, we actually started out with a lower disease severity. I think it takes, they actually kind of need the roots to help them establish. But then, so our first level was pretty low, and then you can see in the March and the May, they're pretty high, and then they went down again in our last one. So it means that the roots, which is what they're produced in, it looks like they need that root to sort of the buffering capacity that actually helps keep them alive. In our controls, we had a bit of a bump here in May of actually having some disease, but I think that's just the natural microbial activity that we see in the soil. There's fusarium and other things in the soil. So Shamila has actually started another big experiment with how many? 120 kilograms of soil that is being incubated at a whole bunch of different temperatures, and that will also follow for as long as we can until we run out of soil, basically. So these results not encouraging here when we see that even after a year, we haven't seen any reduction in those oospore levels. And that's what the plants look like when we pulled them out at the last. So after 9 months and grow them in that soil, that's what they look like. Okay, so like 5 minutes left, so I can get through my last thing, and that is calcium products or liming. And why calcium? This has been really, really well studied as a management mechanism in sugar beets in the US for managing Aphanomyces root rot of sugar beets. And there's been a lot of really great research that's showing that that spent lime reduces the stand loss and root rot in sugar beets, increases yield, sugar content, and really good economic benefit. So we're like, well, if it works for Aphanomyces root rot of sugar beet, different species, very, very closely related, then it should also work for pea. And the reason why, there's some good research in background really showing that calcium is really, really important for some of the life cycle of Aphanomyces. So here we can see over here, very few zoospores are produced when you have a lot of calcium. And then also calcium is required. So oospores, here they germinate to produce the zoospores. The zoospores swim to find the roots. Then the zoospores germinate and infect the roots. And so calcium is needed for that process, but too much calcium actually inhibits the process. So it really is inhibiting two stages of the life cycle of Aphanomyces. So we're thinking, okay, well, calcium is important. This should work. And so in 2021, we just did these microplot trials where we buried a whole bunch of these little tubes inoculated with Aphanomyces and Lyme, and then looked at the disease. What we found is that disease severity for Aphanomyces was actually reduced, although this was not statistically significant. So oospores are pesky, but so are statistics. And so what we found was that, we did see a reduction in disease severity of Aphanomyces, but wouldn't you know it, we saw an increase of disease caused by fusarium. So it's like, well, if Aphanomyces isn't occupying that niche, now we've opened up more routes for fusarium to come and inoculate, just to make our lives more interesting. And then what we saw as well, which again, not statistically significant, is that these blue bars here are Aphanomyces levels on the roots. I mean, to me, it looks like there's a big difference, but the problem is it's so variable. The amounts that are on the roots that are standard errors are so big, so we don't actually end up with statistically significant results. But I mean, this is encouraging to say, okay, it is actually reducing the amount of Aphanomyces that are colonizing those roots. So then we wanted to move from these microplot trials to field trials, which is what we did in 2022. And so we have all of our different Lyme products here. They are applied at very high concentrations. You need to boost up that calcium levels to really high levels in order for it to work. So you can just see kind of how much you have to put on the field. And we are testing different concentrations. And then it is tilled in. That's another issue, that you will have to incorporate it into the soil to have it to work. And what we found is there was no impact on disease severity in 2022. And again, if we're looking at all our sites, Swift Current, Tabor, Redvers, Scott, Saskatchewan, and Lacombe, we can just see like how high those disease severities are. So this is what the roots look like. Like I said, this is about the highest levels of disease we've seen since doing some of these trials. So why, that's what the plots look like in Redvers, like they weren't able to even get any yield off of it, and this is usually one of our better sites. So why like this promise of Lyme? The reasoning behind it seems really sound. It's worked really well in sugar beets. Why aren't we finding the same thing here? I really don't know. Is it because we're dealing with the pathogen complex where we see maybe you're reducing Aphanomyces, but then you're letting fusarium colonize? It might take a while for the effect to actually work. So our plan is to try to follow these soils for a few years to see if we see reduction over time. That is certainly what they saw with the sugar beet in the US. And we don't have the yield and the pathogen levels yet for 2022. But some, like looking at the plots, some of them were like, oh, these look way greener. They have to be healthier. And then you look at the roots and they're not. So there might be something like in the yield that we are seeing. So, this one, this is early phase, first year, so we're still looking at that. So just in summary, to wrap up, sometimes I just feel like I'm the bearer of bad news when I stand up here. And I don't like to be the Grim Reaper. Casey, I think, gave me that name. But 2022 was really kind of an ugly year for root rots. We had a little bit of a reprieve for a few years, and then 2022, it just popped up again because we had those heavy rainfalls right in the middle of June. So really, it's like the rainfall and timing is overriding everything else that we're trying to do. We do see some benefits from either your choice of preceding crop or liming, but only when that disease pressure is low. So drier years, we see kind of better response to some of these treatments. And it's really because these ooze spores are just these pesky little creatures that are here to just kind of disrupt anything we're trying to do. And so that just brings me to, some of the two projects that are just starting. Like I said, we're really trying to delve more into what is triggering these ooze spores. Why do they love peas so much? Why do they know it's a pea crop that's in that field? And if we can figure out what that signal is that's coming from the peas to the ooze spores, can we trick them somehow to thinking that there's peas there when there's not peas? I don't know. And transform oats with a pea gene or something. I don't know. Then we don't have to worry about the P and T issue, right? Anyway, so that's where we're going to. And then, finally, just our overall vision where we're trying to move to with the research is coming together with this cohesive kind of research strategy and target, and looking at, these are the things that we're looking at. The soil test to know what's in your soil and how much, trying to come up with some disease management packages to say, this is how long you need to wait because we know now what the ooze spore decay rate is, and really looking at partially resistant cultivars, which I knew I wouldn't have time. I had some slides in and I took them out because I just knew I wouldn't have time to talk about it. But we for sure are looking at, there's a lot of research going into trying to find some resistance. And so I just want to thank my team. This is actually a 3-year-old Christmas picture, so we need to get a new one this year. And then a lot of the collaborators that we work with in our different trials. And then I love that Jenn kind of used mountain climbing as her wrap-up slide, because I always say my first love is hiking, my second love is Aphanomyces. This is actually just from yesterday when we went up and did a little snowshoe up in Crow's Nest Pass. Anyway, with that, I can take questions if there's time.
Looking at previous imagery.
Yes, so the question was about predicting disease using imagery, because we do know it usually starts in low spots and wet spots. And yes, absolutely. As part of the next pulse phase of the pulse cluster that's coming up, that is a big research activity, is being able to use imagery to detect low spots early, those patches, the early patches where you're just starting to get the yellowing before it spreads to the whole field, is that if we can target those early spots of disease, maybe use lime if we find that it works just in those areas, or use something else, then we can stop it from spreading across the whole field. So that's definitely one of the research plans on the docket.
I saw some speculation online with lentils recently where they said, oh, the non-clear field ones didn't seem to be as impacted. And maybe it was something to do with herbicides going on the crop at the same time as rains are coming. Totally speculation, but is that a possibility? The plant is hurt at the same after herbicide timing from just recovering from that?
Yeah, that question has come up a lot, and I don't have a good answer for it. I would speculate and say that no, that's not an issue, just because of the way, I guess, with the prevalence and the incidence that we've seen across crops, so my primary hypothesis would say that's probably not an issue. The same question has come up about rolling, like, are you stressing the crop and then they're more susceptible to root rot? But I think it's just coincidence that the rains happen to come at that same time as the herbicides and the rolling. So I don't have hard data, but my gut feeling would be to say no, it's probably not a big factor.
Jen is wondering if you could explain how Aphanomyces is different than clubroot, and are there cross learnings?
Oh, yes. So, yeah, tough question, Jen. So when I first started talking about Aphanomyces, I really tried to avoid comparing it to clubroot. Just, I don't know why, I guess just semantics. But yes, it is actually very similar when we look at the life cycle, the fact that you have these resting spores that survive in the soil that make it a lot more difficult to deal with. And a lot of the learnings from clubroot come with sanitation and reducing movement of soil from infested fields to non-infested fields. So when we talk about sort of biosecurity issues, like when we go in and do our surveys, we're, even though we're not clubroot down here, we're like, well, we don't know which field have Aphanomyces, so we're always wearing our booties, we're always bleaching our shovels between fields. So in terms of all that research that's gone into clubroot in preventing movement from field to field, that is absolutely the same for Aphanomyces. And then as well, a lot of these issues of detection and quantification of clubroot spores in the soil, we've looked at a lot of those papers and a lot of the research that's gone into that, to also applying that to kind of the Aphanomyces research. And then liming is actually another kind of big kind of crossover between the two, because there's been a lot of research on liming and clubroot, and that one's more related to pH than it is to calcium. But it just so happens that both of these organisms, maybe there's something with the calcium and pH that really affects their life cycles. So when we're talking about liming, we're particularly thinking, oh, that could be really useful in fields that have both clubroot and Aphanomyces, and like, don't want to think of that. But I think, up around Edmonton, you can certainly come across fields that would have both. Down here, we're at least safe from clubroot, but knock on wood. Anyway.
One last one from Jen. Can Aphanomyces be transferred on seed? Is there enough earth tag actually on a seed?
Yeah, that's a really good question, and one that I get a lot. And I'm fairly confident to say it cannot be transferred on seed. Usually when we see Aphanomyces, you see it stop right at that soil level, so it doesn't move into the upper part of the plant. So the only time you would be at risk is if there was some earth tag, but you'd need an awful lot of earth tag. You'd have to have more dirt in your seed than you would seed in order for it to actually transfer. We've tried for like 5 years to move infested soil from an infested field to our site at Lethbridge to start an inoculated disease nursery, and it just does not take. So you do need a fair amount of soil to actually get an infestation going, and there wouldn't be enough on earth tag related to seed.